All right. Well, thank you, Lauren. I really appreciate that. All right. Well, today I'm going to talk to you about some work that I've been doing over the last couple of years since the pandemic started, looking at novel interactions that the virus has with receptors that we've identified in the body, and then how we can therapeutically target that and the impact that that has on the brain. So our work is really focused on the spike protein. I'm not sure if can you see my pointer as I move it on the
Yes,
screen?
and
Okay,
we can.
excellent. So our work is really focused on the spike protein, which the virus uses to bind to cell surface receptors and ultimately invade cells. And we identified pretty early on in the pandemic, we and others, that the amino acid sequence of the spike protein contained a particular 3 amino acid structure called RGD. This is a particular binding motif in protein structures that recognizes a particular class of cell surface receptors called integrins. And what was particularly interesting about this, twofold. One, that SARS-CoV-2 was relatively unique amongst other coronaviruses that you can see listed on the screen, in that it had this binding motif where these others did not. And it was speculated pretty early on in the pandemic, in early 2020, that the presence of this integrin binding domain on coronavirus spike protein might convey some of the virulence that the virus has unleashed upon all of us. So with that being said, we and others looked at what was happening to this particular spike protein as the various variants emerged over the last couple of years. And sure enough, with each mutation in the spike protein or mutations, this integrin binding motif, this RGD, which was already fairly prominently expressed at the tip of the spear, so to speak, was even more prominent with various mutations. And here you're looking at the UK variant, but subsequent variants have borne this out. So there is a specific receptor binding domain on the tip of the spike protein of coronavirus that conveyed a unique ability to bind to this class of receptors known as integrins. All right. So integrins, very basically, they are composed of 2 subunits, an alpha and a beta, and they bind to something called extracellular matrix. Extracellular matrix is essentially the glue that helps hold cells together. It forms basement membranes. Every cell in the body expresses integrins of different complements. These subunits are referred to as alpha and beta, depending upon which alpha binds to which beta or associates with which beta. You get receptors that have different affinities for different components of the extracellular matrix. We were particularly interested in some that are expressed in the brain. And this schematic essentially shows integrin subunits that help form what's called the blood-brain barrier. I'll be getting back to this in a little bit. But essentially, there's a barrier that isolates blood and vascular compartment from the brain and the brain parenchyma. And this is tightly regulated. And viruses and other infectious agents can impact the blood-brain barrier. And as a result, there can be infection in the brain or swelling, inflammation, that sort of thing. But these integrins that I was referring to help regulate the tightness of this blood-brain barrier, which will become very important in a minute. And then we've spent a lot of time in my lab looking at specific modulators or inhibitors of integrins. And then one that I'm going to talk about a fair bit, something called ATN-161. This is a peptide that can bind to integrins such as alpha-5 beta-1, which we have since identified as a COVID receptor. And we've done a lot of work to look at whether or not this inhibitor peptide might actually have therapeutic efficacy against coronavirus. Right. So this is a summary of a couple of years of work, but suffice it to say, here you're looking at the spike protein of the virus. It interacts with known receptors. ACE2 is one that you've probably heard of, and associated integrins. And this interaction facilitates the virus getting taken up into a cell, and then all the bad things that happen as a result of that host cell invasion. So we've done a lot of work to demonstrate essentially that if one takes just the spike protein and puts it on cells in culture. In this case, these are brain endothelial cells. We looked at a number of different indicators of blood-brain barrier integrity, and we see that the spike protein by itself can actually change the expression patterns of these proteins associated with blood-brain barrier. It reduces them, which is a clue, and since validated by others, that the virus can actually impact vascular barriers. We did a lot of work early on in the pandemic to essentially show that these spike proteins can bind to integrins, and that we could block this interaction by co-incubating with this peptide I referred to before, ATN-161. This was ELISA studies that were done pretty early on in COVID-19. We showed, I think equally as importantly, that if we added cells, the virus rather, live virus to cells, we could block a lot of the viral replication by adding this inhibitor to alpha-5 beta-1 integrin. That's shown by graph here. And here you're just looking at the cells that were co-incubated with virus. All these little brown, round balls or plaques are cells that are impacted by the virus. When we treat it with our ATN peptide, we can largely suppress these round cell formation, which is synonymous with viral replication. So in animal models, there are a number of different animal models for looking at COVID-19. My lab's taken advantage of a number of different animal models as they become available. In one particular model, you can make a normal lab mouse express the human form of ACE2, which is a known receptor for the virus. At the time, this was the only way to make a standard lab mouse susceptible to infection. If we inoculated these mice with coronavirus and then treated them with ATN-161, we could significantly suppress viral load in the animals. And in a different model where instead of making the mice express the human ACE2 with the virus, we could actually use a transgenic mouse, something called K18, that expressed human ACE2 everywhere. And similarly, if we infected those mice, we could actually block the infection by treating with ATN-161 as well. But a better tool that emerged in the last year or so is a version of coronavirus that was mouse adapted. So this was a SARS-CoV-2 that was passaged in standard laboratory mice, put into a mouse, allowed to infect, taken out of the mouse, passaged into a different animal, et cetera, et cetera. After 10 passages, the virus could actually infect wild type mice. This really facilitates coronavirus research in labs because now one can work with standard mice that most labs have access to. And so we took advantage of that. And we did a number of studies, and when we infected mice, just and waited 2 or 3 days and started looking at markers in the brain, we noted that those same tight junction proteins, these are proteins that stabilize, facilitate the tightness of the blood-brain barrier. Here you're looking at Claudin 5 is one. We see a significant reduction in the levels of this protein. Again, that's important to blood-brain barrier stability. We also see a significant increase in inflammatory cells and astrocytes in the brain with coronavirus infection. Now, I should point out the virus is put into the lungs. So we're looking in the brain 3 days later. And this is in the absence of virus actually getting into the brain. So we're seeing changes in the brain that indicate blood-brain barrier disruption and neuroinflammation 3 days after infection. This is just another example. We're looking at a stain from microglia, which are the brain's resident immune cells. There's a significant increase in immunoreactivity of microglia 3 days after coronavirus infection in standard mice. So this is just another schematic, again, where we infected mice, standard lab mice with MA10. And then we also treated with that ATN-161. And very nicely, we saw effects on various markers of inflammation. This is interleukin-6, pro-inflammatory cytokine in the brain. We saw that ATN-161 could significantly reduce the pro-inflammatory cytokine in the brain. We saw similar effects on blocking the reduction in Claudin-5, which again is a marker of blood-brain barrier integrity. Very briefly, I think everyone on this call is familiar with long COVID, so I won't belabor this slide. But suffice it to say, one of the changes that have been noted in clinical long COVID is a reduction in blood flow to the brain. And there's a number of different reasons why this may occur, but we decided to model this in those standard laboratory mice by putting coils on the carotid arteries. These are major blood vessels that feed blood to the brain. And if you restrict but don't eliminate the blood flow, typically what happens in a standard mouse is after a couple of weeks, there's white matter injury in the brain. And after about a month, these animals begin to develop cognitive impairment. So it's a chronic hypoperfusion of blood to the brain, resulting in a cognitive decline, a so-called vascular dementia. We did a number of studies that essentially showed that if you do this, wait about a week after you put these coils on and then infect the mice, their brains are much worse than if you just infected them alone or if you just put the coils on by themselves. This is alpha-5 integrin, which is a receptor we'd identified for coronavirus. Levels in the brain go through the roof. We saw changes in microglia. We saw pretty significant changes in GFAP, the astrocyte marker, again, in the brain. I'm going to skip ahead. Essentially, if one looks months later after a infection in laboratory mice, we see clear indications of neuroinflammation that are chronic. So this is 30 days post-infection. We see changes around blood vessels in the brain that persist in experimental long COVID. And we see significant dropout of neurons in parts of the brain where you don't want neurons to go away. In this case, the hippocampus, which is important for the consolidation of new memory into long-term memory. This is 60 days after infection. In a number of the mice, we saw significant neuronal cell dropout. And this is just showing signs of inflammation again that were chronic 2 months after infection. So I want to leave you with this idea that we're now at the stage where we can nicely model rotavirus infection in standard laboratory mice. And unfortunately, in a number of these animals, days to weeks to months after infection, we see changes in the brain, which are made worse when we superimpose reduced blood flow, when we model cognitive decline. And however, we think there are particular receptors that make coronavirus particularly virulent that we can also modulate, in this case with ATN-161, which we are also studying as a potential dementia therapy. And I think because this receptor may play a role in both dementia and vascular integrity and coronavirus infection, it may serve a dual purpose in both treating the virus as well as the vascular sequelae associated with it. So I think I hopefully haven't gone too far over. I'll just acknowledge the team that helped do the research and my collaborators, and I guess we'll wait for the very end of your questions, but thank you very much for your attention.